6 on microbial nutrition. The topics for this first set of slides is nutritional types and looking at new vocabulary terms to describe microorganisms and their nutrients that they need for carbon and energy, and then looking at nutritional transport, how organisms bring things inside the cell. There are 4 key concepts for this chapter. The first 2 in this first set of slides are understanding tonicity. This is also known as osmotic pressure, or the movement of water into and out of the cell, depending upon the environmental and intracellular solute concentrations. And the second key concept is passive versus active transport. So first, the definition of microbial nutrition. Nutrition, nutrients acquired from the environment and used in metabolism and growth. Now, if you are following along, there we go. These 2 terms mean 2 different things. So for metabolism, this is what we'll talk about in chapter 7. This is looking at the use of organic compounds or inorganic terminal electron acceptors to generate energy in the form of ATP. Usually, most organisms require glucose as a starting compound, or peptides like amino acids or lipids. So these are all organic compounds. Some organisms can utilize some of these things and use an inorganic substance to actually generate energy in the end. This is as opposed to growth, and growth is more biosynthesis, right? So we're looking at synthesizing biological components. And when we get into our next chapter, you'll see how these relate to these terms. I'll just write them down here. So these are chapter 6, nutrition notes. And I will make these notes available. I don't know if I've had that in the past, but I will make these available on Canvas. So our basic definition here of nutrition is nutrients acquired from the environment and used for metabolism and growth. And again, I just wanted to specify when we're using this term metabolism, this is going to equate to a term called catabolic processes or catabolism, which is where nutrients are broken down to produce energy. And we'll talk about this in chapter 7. And this is as opposed to this term we've used growth. In this case, we're talking about anabolic reactions or anabolism, which is biosynthesis. Biosynthesis, or building using macromolecules to build cellular components. And this one is also an exergonic reaction, exergonic, meaning the energy is needed as input for growth and energy is released as output for catabolic reactions. So let's go back and look at some more concepts here. So again, nutrients are needed in this case for growth, for synthesis of all cellular components. We're talking about everything we've talked about in the last few chapters. So cell wall, cell membrane, proteins, nucleic acids, etc. These are all derived from the 4 basic building blocks of life, including fatty acids, carbohydrates or sugars, amino acids, and nucleotides. And these have basic compositions, carbon, nitrogen, etc. And they're all derived from diverse sources. So many of these building blocks can be synthesized by the cell. I'm going to see if I can get this pen to work. No, not yet. Whoa, sorry. Playing with my new pen. Many can be made by the cell, but many have to come in from outside sources. So the nutrient source comes in, and let's see. The nutrient source nutrient source comes into the cell. The cell will usually break down that nutrient source to get required elements or macromolecules that can be used for either catabolic reactions or biosynthesis. As mentioned, they can be used, so there are the building blocks. Here's fatty acids, carbohydrates, nucleotides, and amino acids. And these can then be redirected to biosynthesis reactions. For example, fatty acids are needed to make that phospholipid bilayer. Nucleotides, of course, are needed for making nucleic acid like DNA and RNA. Amino acids are required for making proteins. Carbohydrates can actually go into biosynthesis reactions. So many obviously are peptidoglycan, or the bacterial peptidoglycan is made of carbohydrates and peptide linkages. But also carbohydrates are really important in producing the energy needs of the cell. This is a picture talking about some of the required elements that the cell needs to obtain from the environment, the major elements and the trace elements. Most of the trace elements are required as cofactors for enzymes. So if the cell does not bring these in from the environment, or alternately, if the environment does not have a substantial source of these trace elements, then the cell will not be able to function because the enzymes won't work, and then that will halt metabolism. This is a better illustration or image to capture the same concept here. This is a table showing the chemical composition of E. coli in dry weight. That kind of just means if you were to powderize, lyophilize, remove all the water, and then take all the individual components. This is a decent picture, or a good way to evaluate the contribution of each of these different compounds to the cell. So you can see the majority of the cell is made up of proteins, right? And this is not just structural proteins like we think of muscle proteins, but this is also enzymes. And this is actually the majority of the proteins that you find in the cell are enzymes, ones that are required for metabolic and reproduction and biosynthesis reactions. The rest of the stuff, nucleic acids, carbohydrates, lipids, and then other miscellaneous items are in that order. Inorganic compounds, water is not included because this is dry weight, but other things make up about 3% of that cell. And then if you were just to look at breaking each of these things down into their actual chemical element, carbon is, in all living organisms on the planet, is the highest or the most prominent compound or element, making up over 50% of the dry weight of the cell. Oxygen, nitrogen, hydrogen, and phosphorus. These are the 5 really key ingredients to all living things on our planet. Here's the rest of the stuff: sulfur, potassium, sodium, calcium, magnesium, chloride, iron, and trace elements in very, very small amounts. Right? But again, all of these things are very important. Just because they're in small amounts doesn't mean that they have less significance. Without these trace elements, including these smaller micronutrients like iron, then you would not have cellular function. So here are some terms for how we characterize bacteria based on their requirement for, or their source of carbon and their source of energy. Now, trophism or trophy is a term that's related to growth requirements. So we've seen this term before when we were looking at halotrophs, right, in the laboratory, where we were talking about the ability to grow in the presence of, in that case, halo would be salt. And so these are different prefixes that you can add to that term, troph or trophism or trophic, to indicate some type of requirement for growth. Now, when we're discussing the source of carbon, we're going to equate this to our biosynthesis requirements. That's not exactly true, but we'll go back up and add this to our little list here. So for growth, we're going to also add that this is our source of carbon terms, and that would include auto and hetero versus the source of energy here. In this case, we're really going to focus a little bit more on the ability to use different types of compounds as either the energy source or the terminal electron acceptor, which we'll talk about in chapter 7. So here we have chemolitho or chemoorgano or photo. And for energy, again, we're going to go back up here to metabolism and say that we're going to talk about our source of energy terms. And again, this would be either chemolitho, and this means using inorganic compounds. Chemoorgano, which would mean, again, these are prefixes, which would mean the use of organic compounds. And an organic compound means that it has carbon and hydrogen and other things, usually carbon, hydrogen, and oxygen, but at least carbon and hydrogen. And photo means, of course, as you would imagine, they use photosynthesis. Sunlight, yeah, photosynthesis. Sorry, let's go and add into these terms down here for our source of carbon. Auto means they're photosynthetic, so they can fix carbon from CO2 using photosynthesis. Right, fix means they take it from that gaseous form and put it into a chemical form. Everyone realizes photosynthesis or remembers this from general biology, uses carbon dioxide and sunlight and water to make glucose, which is C6H12O6, and glucose is then stored as cellulose in the plants, which is plant biomass. Hetero means they use other organic compounds. So let's look at these terms here just one by one. Again, autotroph means the source of carbon is carbon dioxide. Please don't get confused and don't describe an autotroph where their source of carbon is themselves, right? Even though auto means yourself kind of thing, it's definitely not. You can't generate carbon from nothing. So carbon is from carbon dioxide. Through the process of photosynthesis, it's fixed into glucose. Nitrobacter is one example of a photosynthetic bacteria that is also an autotroph, able to generate its own carbon from carbon dioxide or generate its own chemical compounds. The next here is a heterotroph, and this is where pretty much every other organism falls into this category, including us. Heterotrophs means they don't undergo photosynthesis, and therefore they have to get their carbon from organic compounds that they acquire from their environment. So their nutrient source must be some organic compounds to get the carbon. It's a picture of Pseudomonas, which is a really common heterotrophic bacteria. Now the other terms here are for energy. Those two are for carbon, and this is for energy. For energy, organisms can get their energy through photosynthesis, so using sunlight to generate energy. This is a picture of cyanobacteria, which is another photosynthetic bacteria found in aquatic regions. Chemolithotrophs are those whose source of energy is an inorganic compound, like these halothiobacillus, purple sulfur bacteria. These guys get their use sulfur, a sulfur compound like a sulfate, as an end electron acceptor, so that they can use something called anaerobic respiration, which we'll talk about in a lot more detail in the next chapter. The most common or the biggest group of organisms classification would be chemoorganotrophs. These require organic compounds like glucose as their starting energy source, and they usually undergo the process of fermentation or aerobic respiration. And then you can put these terms together, right? So we use terms like photoautotroph to describe photosynthetic organisms that are both able to get their energy from sunlight. So energy would be the first term we use, and then carbon from CO2, which would be the second term we use. And this is a really common category of photosynthetic organisms like plants. Chemoorganoheterotrophs are pretty much most other organisms on the planet, but there are definitely exceptions to the rule. Most bacteria do fit under this terminology. In chemoorganoheterotrophs, the energy, that first term, means they get their energy from organic compounds. And in this case, the same thing for their carbon source, also from organic matter. So these questions you should be able to answer. We'll just go over them here. What is the carbon source of a photoheterotroph or chemolithoheterotroph? Well, you have to remember again that we can group these terms together, but we always do energy first and carbon needs second. So in this case, they get their energy from sunlight and they get their carbon from organic compounds. For this one, let's break it down again. First term is chemolitho, which means they get their energy from inorganic chemical compounds like sulfates or nitrates, but they get their carbon, heterotrophs, means they get their carbon from organic material, right? So both photoheterotrophs and chemolithoheterotrophs get their carbon from the same nutrient source from organic material. Now you may be kind of scratching your head, is it possible for a photosynthetic organism to not get their carbon from photosynthesis too? Yeah, it definitely is possible. So many bacteria are able to kind of do both. They do have the ability to undergo photosynthesis, but they also can undergo standard processes like aerobic respiration, pulling in a I'm talking about biosynthesis. They can also undergo standard biosynthetic processes where they still need to bring in these macronutrients from the environment and use them to get their carbon. Same thing here. Chemolitho means inorganic compounds for energy, and then heterotrophs means organic compounds, and this is definitely true too. So if an organism can undergo something like anaerobic respiration using nitrates or sulfates as a terminal electron acceptor, and again, that's a lot of words, but we'll talk about that in a lot more detail in the next chapter. They still need some carbon source to get their carbon from. So chemolithos don't necessarily need carbon for their energy source, so they do need to still bring in organic material for their carbon source. This next question here, which of the following might acquire energy from photosynthesis? Let's answer that question first. If you look at the list here, energy, remember, is our first term, and the only one that shows the ability to undergo photosynthesis is the first one, photoautotroph here, which is likely found in the human gut. This is a great application question. You first have to think about the environment of the human gut, that's us, and our diet. So what nutrients will be present in our gut? Well, it's mostly based on the stuff that's present in our diet. We are chemoorganoheterotrophs. We require organic chemical compounds for both our energy source and for our carbon source. Therefore, you would probably expect to see the exact same things in our gut, chemoorganoheterotrophs. Definitely unlikely to find photoautotrophs, except accidentally perhaps as a contaminant going through your system, but there's no sunlight, so we really don't have the ability to populate our stomachs, our guts with photoautotrophs. And then chemolithoheterotrophs, definitely heterotrophs. And then do we have any inorganic compounds in our diet? Well, likely yes, unless we live out in the bushes of sub-Saharan Africa or something where we don't have preservatives. Most American diets have preservatives that have inorganic compounds in it, and therefore we probably have the ability to encourage the growth of these types of bacteria. And actually, I will say not only from a dietary perspective, but we also have evolutionarily adapted, or I would say we have sort of created these monsters, these pathogens that are able to utilize some of the defense system that we have in place to eliminate pathogens. They have kind of learned how to use those things to their benefit, and there are some bacteria, for example, that can use the nitric oxide that we secrete out that's really, really harmful to most bacteria, and they have evolved to be able to turn that compound into a usable nitrate and use it for their energy source. So in that case, they're using the host defense system to actually thrive. It's quite phenomenal. All right. So let's talk about transport, how organisms get stuff into and out of the cell. And to begin, we'll look at the movement of water. This is the process of osmosis. Now, in class, I drew this on the board, but since we have the screen in front of us, I'll just kind of highlight what's happening here. So let's look at this illustration of a beaker that's filled with water. In the center of the beaker, there is a semi-permeable membrane. This membrane has pores that allows the movement of one of the solutes here, but not the movement of the other. So you can see that the blue guys can move freely back and forth across this membrane, but the red guys cannot move across this membrane. So this membrane is impermeable to the red guys. but it's permeable to the blue ones. Now, in all things in biology, it's based on chemistry, so all things in chemistry, you'll find that all things tend towards isotonicity or equilibrium, right? And so that's no different here. Isotonicity means that you'll have essentially the same concentration of solutes on either side of a membrane. Now, if you look at the blue guys first, you'll see that in this illustration we have pretty much equal concentrations of the blue guys on either side. And how do we know this? Well, I mean, you could count the number of molecules on one side versus the number of molecules on another. But we're going to focus a little bit more on the concept of the density. So how dense something is really how close two molecules are to each other. And if you just kind of look at the blue ones, we see that, suppose initially that the blue guys might have a little bit more space here, but over time the density is going to be exactly the same. So concentration will reach equilibrium as the blue guys passively diffuse across that membrane. Now for the red ones, this will never happen, right? Because red cannot diffuse across the membrane. But we know there's one other component here that can pass through the membrane. So we have the blue guys and the red guys and water. So water can move even if the solutes can't. And in fact, water will move when there is impermeability to the solutes, so that the solutes do become equal on either side. Now in this case, we'll have to figure out which direction water moves. So if the concentration is lower here, and again, you could count molecules, but again, a concentration is not about how many molecules there is, but how many molecules per, volume in this case. So we're really talking about the density, or how closely together these guys are in this aqueous environment. So you can see that these guys are much closer together than these ones here. So water needs to dilute out the higher concentration, or the volume here needs to increase so that these molecules become further spaced apart, right? So water will flow from an area of low solute concentration to an area of high solute concentration in order to diffuse out the high solute concentration. So in this illustration, we'll see that water moves from the left side to the right side. And so here it is, water moved from the left side to the right side, making the volume on the right side higher, which essentially dilutes out or makes more space between molecules here, which essentially reduces or lowers the concentration of the molecules without having an effect on the number of molecules on either side, right? The red guys are still the same, but the space between them is greater here. And the space between these guys got smaller, which makes the concentration higher, so that the density became higher, the concentration became higher of the solute on this side. So again, water will passively diffuse. Diffusion of water across the selectively permeable membrane is called osmosis. It's a form of passive transport, and it does follow a particular pattern. So it follows solutes. It follows the kind of the inverse of a concentration gradient. So we'll always move towards a higher solute concentration. This term I've been using to talk about the passage of things through a membrane is permeability, or something is permeable, or the membrane is permeable to a solute or to something. So if the membrane is permeable to a molecule, then we call that passive or simple diffusion. And this will always flow with the concentration gradient. That means that it will go from that solute will go from a high concentration to a low concentration. The speed and direction of that diffusion action will be based on the relative concentration on either side, which means if you have a very significant difference in the concentration. Let's see if I can... So if, for example, we have here we go. If, for example, we have that same illustration with that selectively permeable membrane, and we have a significant concentration gradient here, where you have a lot of solutes on one side and a very small number of solutes on the other side, and we'll say that this membrane is permeable to these little molecules, then we have the rate and direction is determined based on the concentration gradient. So in this case, we have a very significant gradient, only a very small number of solutes here, a high number of solutes here. So the direction will be from a high concentration to a low concentration, and the speed it can actually be measured, but it will be a very rapid rate of diffusion because of this significant gradient. That would be, of course, opposed to something where you still do have a difference in concentration, higher concentration here, lower concentration here, but if the concentration difference is not much, again, speed and direction will still be determined based on the relative concentration on either side. So in this case, it still will move from left to right because the solutes are still higher here and lower here, but this will be a much slower rate of diffusion because the concentration difference, the difference in that gradient is not very significant. Now again, the movement of water is called osmosis. And here's a picture of everything we just talked about using that gradient difference, using that selectively permeable membrane. But now let's look at that instead of in a beaker with a membrane that you put in. Let's imagine that membrane is now the cell membrane, which again is the same concept. It's selectively permeable. In fact, it prevents the passage of most molecules freely back and forth and requires transport proteins instead. So more often than not, you will have something like this where the solutes are impermeable, or I should say that the membrane is impermeable to the solutes, and therefore there is a concentration gradient that becomes established on the inside and the outside of the cell of the solutes, and water adjusts for that concentration gradient to create isotonicity or equal concentrations on either side. In this picture here, we have a low amount of solutes on this side of the membrane and a high amount here. And remember, our rule is that water will flow from an area of low solute concentration to an area of high solute concentration. Here's a picture of two different scenarios. Here on the left side, on the top, is a scenario where the concentration of solutes is higher inside the cell than outside the cell. So water again will flow to the area of high concentration to dilute this out. This results in the swelling of the cell. The only thing that prevents the cell from lysing in this high osmotic pressure is the cell wall. And even that, when the concentration gradient is pretty extreme, the cell wall cannot prevent the bursting of the cell. Here's the opposite of that, where we have a low solute concentration on the inside and a high solute concentration outside, making the water go from an area of low concentration to high, which again would mean it's flowing outside of the cell, and in this case the inside of the cell becomes dehydrated or it shrinks, crenation, etc. Here are some vocabulary terms for you to describe each of these environments: isotonic, hypotonic, and hypertonic. And we'll see how each of these things are played out. An isotonic solution is one in which the solute concentration is equal on either side of the membrane. In isotonic environments, which actually is quite rare in biology, but in isotonic environments, you would have free movement of water into and out of the cell at equal rates, right? Nothing in biology is static, so you would not have a cell that doesn't have water moving, but you would have free or equal movement of water both into and out of the cell. Even if the cell is lacking a cell wall, nothing would happen to the cell because you are in a equilibrium with the environment. However, a hypotonic environment, and we're going to use these terms to describe the outside as compared to the inside. So the outside, hypo means under or less. So the hypotonic environments are ones in which the solute concentration is lower outside than inside the cell. Again, water moves from an area of low solute concentration to high solute concentration. Therefore, water moves into the cell here. An example of this environment is a fresh pond water or any aqueous solution that is not salt water. And you can think of this simply as salt concentration, although that is definitely not the only solute we are discussing here. In fact, glucose, carbohydrates can also create these various tonicities. But in the fresh pond system, organisms that can live in these fresh pond waters must have evolutionarily adapted to that environment in some way. The result of a hypotonic solution is the swelling or potentially bursting of the cell. So we know already that the cell wall is important in preventing the cell from bursting, but also some organisms have physically altered some property of the cell, like the amoeba has essentially a water pump or a water vacuole that pumps excess water out of the cell. So although they continuously face influx of water, they actively pump it back out again. This is from your book to show this hypotonic solution. Again, higher solutes inside than out, or lower solute concentrations outside than in. In these environments, water is flooding into the cell, but the cell wall can prevent that cell from bursting. If a cell does not have the cell wall, which may be the case in the presence of an antibiotic like penicillin, for example, or there are other organisms that can exist in what we call the L form that does not have that peptidoglycan. But in that case, or in this circumstance, the flooding of that, the movement of water into the cell causes the cell to lyse. Hypertonic solutions are the opposite. Hyper means over. In this case, there are more solutes outside the cell than inside the cell, and water will move towards the higher solute concentration, so will leave the cell in order to create isotonicity. An example of this would be a high salt environment. Organisms that are adapted to live in these environments must either restrict water loss or must increase their own internal salinity, so make themselves saltier. A great example of this is the halobacter or halobacteria, an organism that lives in the Great Salt Lake, which is, I forgot what concentration, something like 10% salts or something higher than that. Crazy high amount of salt concentration. So there are organisms, extremophiles, and other bacteria that can live in here, and this organism does this example here, so it increases its own salinity to become isotonic with its environment. A picture from your book to show you what happens here. So again, in these hypertonic solutions, water will be leaving the cell. The result of this is the cell structure still stays the same because we do still have that cell wall here, but you'll find the shrinkage of this internal contents. So the cell membrane will kind of shrivel up here. The cell will become dehydrated. You'll find the shrinkage of the cell, or crenation, desiccation. All of these terms kind of mean the same. Plasmolysis is another term that you can use to describe what's happening here. And it really doesn't matter whether you have a cell wall or not. In the absence of a cell wall, you still find the same thing. So here's a discussion question for you to think about this environment. Explain what happens to resident skin biota when you take a freshwater bath. Assume they could adhere tightly to your skin. What type of environment is the bath? Which direction is the water moving? What cellular structures are involved? And what happens to the organisms? In this discussion question, you really just need to think about all of these scenarios. So freshwater bath, you should think back to those different examples we just gave, and we know that this is very similar to the pond water, which means there's low solutes here. So therefore, it is a hypotonic environment. Bacteria who are resident may have evolutionary adaptation, but likely they still take on, they have evolutionarily adapted to our skin, which is very salty, so they probably have a higher solute concentration inside them anyway. And when you put these guys in this hypotonic environment, then you can start thinking about what would be the result of this scenario. Let's pause here for a second. Okay, so let's talk about different types of transport. So the way that cells get stuff into and out of the cell, passing through that cell membrane. In fact, let me see if I can get this sketch going. Maybe. Maybe not. Okay. So, whoops. Let's see if I can get our something going here. Okay. Let's just draw a really large picture of the cell membrane here. And I'm sorry, I don't know why, but whoops. I don't have my, there we go. Okay. Here's our cell membrane. Now, remember this is phospholipid. I'm not going to go through the problem of drawing the phospholipid bilayer, but this is our cell membrane. And let's talk about how things move back and forth. So things like water, we've just talked about, H2O, and oxygen gas and carbon dioxide, other small molecules. These things can passively diffuse into and out of the cell, okay? This is a form of passive diffusion. So up here on the top, we'll put passive. Passive diffusion. And this is one example of passive diffusion. This would be osmosis, of course, is what we just talked about for this one. Or the movement of other small molecules would simply be passive diffusion. And we just had some rules for these things, right? For osmosis, we know that water moves from an area of high solute concentration to low solute concentration. For passive diffusion of other molecules of solutes, then we know that these things move from an area of low solute concentration to high solute concentration. Nope, that's the opposite. Sorry. See if I can erase that. Things move. There's my eraser, I think. That was bad drawing anyway. Okay. Let's redraw that. Okay, again, so for passive diffusion, things move from an area of high concentration to an area of low concentration, and there is no requirement for transport proteins for this example here. Now, most other molecules, when they can passively diffuse through, they actually require a transport protein. So this is an example of a transport, that should say transport protein, or a transmembrane protein. Try in here. All right, a transport protein, and these are transmembrane. Trans means spanning or across, and the membrane, of course. These are proteins that are embedded in the membrane, and they have a high affinity or particular binding recognition to specific types of molecules. So if the cell can bring in, I don't know a good example of this, but let's say that this is able to freely pass through from outside to in, perhaps something like a chloride ion, and it comes in with its concentration gradient. It's still a passive transport, but in this case, because it requires this transport protein, this is now called facilitated. Facilitated diffusion. Again, it's still diffusion because these guys still move from an area of high concentration to diffusion to low concentration, but it's facilitated. It requires a transport protein in order for this to occur. Now, these are in contrast to a different type of transport, which is active transport. Active transport. Now, two key features of active transport: always requires a transport protein. and always requires energy. Okay, so the most common form of active transport is the movement of molecules across its transport protein from an area of high concentration. Let me give you an example that you'll see a bunch of in our next chapter. Let's say there's a high concentration here of these ions, and they're passed through. That is, oh my gosh, I'm sorry, guys. Always requires a transport protein and requires energy. Now, I do want to mention also that this energy can either be in the form of ATP or potentially even through reduction oxidation reactions. So keep that in mind. We'll talk about this more in chapter 7. But in any case, here's our transport protein. A good example of this is the proton motive force, where protons that are in low quantity here and actually are in high amounts in the periplasmic space here are actively pumped out with the assistance of these proton pumps and requires the input of energy. So I'll also kind of say that energy is required. In this example that I'm providing for you, the energy here is in reduction oxidation reactions, so you'll actually see electrons being passed back and forth in these transmembrane proteins, but we'll get into that in the next chapter. For now, just note we're talking about moving something from an area of low concentration to an area of high concentration. And I will mention there are a couple of conditions that you might see a cell undergo this type of active transport. One might be exactly this, where they need to pump things, move molecules against concentration gradient, which means, like we see in this picture, move them from an area of low concentration to an area of high concentration. However, that's not the only time the cell would use active transport. Perhaps it will move with the gradient, but it needs it faster, but at a faster rate. So those are two circumstances that a cell might use active transport, but in all cases, these two are always true. There's always the requirement of a transport protein like this, and always an input of energy, either in the form of ATP or through reduction oxidation reactions. Now there's another type of active transport that is also quite unique. This is called group translocation. This is really commonly done with glucose, and there are other examples of this, but glucose phosphotransferase system is really, really common. Glucose is also actively pumped into the cell, and in this case, during the passage of glucose into the cell, there are other enzymes involved in this reaction, such that when glucose, or whatever example is we're talking about, comes in, it has been chemically modified. And in fact, in this case, since I'm doing glucose as an example, this is modified in the form of glucose 1-phosphate. So there is, in this circumstance, there's an input of energy in the form of ATP. Must go into this reaction. And because there are multiple enzymes involved in the transport of this protein, we call this form of active transport group translocation. I'll put that down here. Group translocation. So it moves something, translocates it from one side of the membrane to the other. It involves a group of enzymes, and in this case, the molecule, transported molecule or compound, is modified during transport. All right, so these are both two different forms of active transport. And then this last thing that I'll write on here is another type of transport that bacteria cells, I'm not even sure if they really undergo this very much, but this is called endocytosis, and I'll keep it here because I think your book does talk about, whoops, sorry. Let me undo that. I don't know what just happened. Oh, these are my layers. Okay. Okay. Anyway, group translocation. And this last one here is where the membrane itself folds in. Sorry, that's a horrible picture. Here's the membrane. And this is usually if there's some large molecule that is pulled into the cell through the process of endocytosis. This also requires energy to do this process where the cytoskeleton folds in on itself. And you can consider that part of active transport, but it's quite a unique process. Okay, so going back to these transport things, everything we just talked about, you'll see in these slides, but I just have that visual illustration for you. So we just talked about passive transport as a diffusion of molecules from an area of high concentration or density to low concentration. And remember that this is dependent upon the selective permeability of the cell membrane. So only very specific types of molecules are able to passively diffuse across this membrane. If, however, molecules move across that, diffuse across the membrane from high to low without the input of energy, but it's mediated by a transport protein, we call that then facilitated diffusion. Here's a picture from your textbook of this process of facilitated diffusion, where here we have a high concentration outside, low concentration inside, and these are allowed to passively diffuse through with the assist of this transport protein, right, to go to the inside of the cell. And I will just mention that all things in chemistry, this isn't like a tunnel that, you say the magic word and something goes through. Everything here is, everything in biology is based on chemistry. So when you see an image like this, just know that there is a chemical interaction. This protein is made up of organic material, carbons, hydrogens, oxygens, phosphorus, maybe some other minerals like trace elements, iron, etc. And these all have a particular affinity or binding preference for specific types of molecules. And if there is that binding will mediate a conformational change in this transmembrane protein that allows the passage of that molecule into the cell. If something is not able to passively diffuse through, as mentioned, if the cell needs to move a molecule against its concentration gradient or with the gradient but at a faster rate, then it will actively transport things through the input of energy. And remember that in all cases will require a transport protein. So here's a picture exactly like what I drew, much better of course, where protons can be actively pumped out of the cell, or at least into the periplasmic space here, where there already is a very high concentration of that molecule, but in order to do so, this requires the input of energy. In this picture, it's showing energy in the form of ATP, but we'll see in chapter 7 that this is the reduction-oxidation reactions. Another form of group of active transport is group translocation, as mentioned. This is where a molecule is chemically altered upon passage into the cell. The example that I gave to you is the one that's illustrated here, called the phosphotransferase system, wherein glucose is chemically modified upon passage into the cell by a group of enzymes that phosphorylated on this passage in. Here's a picture also of another example, very similar to what I just showed you, where a molecule is passed into the cell through this transport protein, and there are assisted enzyme reactions inside here so that that molecule is modified chemically once it comes in. And again, this is ATP driven, so it's another form of active transport. And then the last one that we just mentioned is endocytosis. Herein, this is showing you a eukaryotic cell, and this process is really common in eukaryotic cells, but in prokaryotic cells, not so much. Here you have the invagination of the membrane, so the membrane itself just folds in around some kind of large molecule, and always this is for the purpose of digestion. You can look at this video on your own. It's on our Canvas page and in your textbook. So some questions: can you argue that group translocation is a form of active transport? We've answered that already. I think yes, because our two rules of the road for active transport is requirement of energy and requirement of transport proteins, and both of those conditions are met with the group translocation. What are two requirements? As just said, two requirements are the input of energy and the need for